And yes, again, I welcome you all. So Sneha, over to you. And just few instructions before we start. You can post your queries during the webinar if you have any in the Q&A box which is given below. And then we can take it ahead at the end of the session. So Sneha, over to you. You can take the session.
Thank you, Ankita, for such a nice introduction. So, good morning, all. Myself, Sneha Singh. I am working as an application specialist in Anchrom from last 4 years. Today, I will be sharing with you all my experience and some case studies of HPTLC instrumentation in pharma application. So, hope I will be able to cover all the insights and my knowledge in this session. So let me share my screen. Before sharing, I want to tell you that we will be taking question answers at the end of this session. So I request all of you to post your question and answers in Q&A box so that we can answer your query. So let me share my screen. So this is about today's topic. So today's topic is about majorly about HPTLC and how HPTLC can handle pharma applications. So data interpretations of our pharma
Sneha, can
analysis.
you, sorry to disturb you, can you do it in slideshow?
Yes, just did.
Please share your screen again. It is not visible.
Now it is visible, Ankita.
It's not in slideshow.
Now it is.
Yes, you can go on.
Okay, thank you. So today's topic is about HPTLC instrumentation and its data interpretation in pharma applications. So this webinar is specially prepared to be designed in Zoom and Google platform. Now, before going to the application, let's brush up our knowledge uh, with the basic of HPTLC and TLC instrumentation. So first, I will start with the evolution, how HPTLC has evolved from an TLC to instrumental TLC, and now the generation has been changed. All the instruments are going to be a robotic. So at the end of this presentation, we are going to see how and robotic HPTLC will work in future. So from 1960, we started doing a TLC.
Sneha, your slides are not changing.
There is a network. Now it is, you can see. Okay. So evolution of HPTLC has started since 1960. So in 1960, we started using TLC. We just used to spot our standard and sample, and then after development, we would see it is getting developed, and then after development, we would just see and compare the TLC things. But then in 1980, what the scientists came that we are using TLC for so much, but we are not getting proper results, and we cannot rely on the results. So they started in 1980s, they started using instrumental TLC so that they can get a reproducible results. Then afterwards, they came to the knowledge that one software should be there to control all the instrument which are there in TLC, as it was a modular system and different instrument was used for different purpose. So in 2000, WinCat software came, and now this in 2016, the software has been changed fully, and it has regulatory compliant, GLP compliant, and also it is a pharma compliant software, which is a Vision Cat software. Its version is 3.0. Then slowly after the software and instrumentation GLP compliant came, then in 2016, scientists came to know that as we are using the software and instrument, why not we inculcate a HPTLC methodology which will be using throughout the world, the same thing, same application pattern, same distance. So scientists came to know that a methodology should be coming. So in 2016, that methodology came, and it was officially accepted in the pharmacopoeia in 2018. And then slowly a regulatory compliant came with it. And then now, that is from 2021, we are using robotic HPTLC in our lab at Mulund Anchrom. The first robotic HPTLC Pro was installed, and since 2025 we will be full-fledgedly installing all this instrumentation for QC department. We will be using this robotic HPTLC for QC department. So now the generation has been changed, and it will be a robotic HPTLC. There will be a modular system, but it will work fully automatic. So this was the evolution of an HPTLC, which was started from a TLC basic kit. Then now what we are having and what we are going to do, that is a robotic HPTLC in future. Then next, TLC. So this was the TLC which we were used to do. We used to spot our standard, then sample. Then after development to certain distance, we come to know that this is our standard. So what we will do the other things like HPLC and all. We will do, and we will confirm. But the HPTLC is this, what we are doing now, or what we are doing since 2000 series. So this is HPTLC. We are applying it in band. So band process is there. This is a spot process. It is fully sprayed onto your plate. The process of TLC was the contact process. So in contact, every person has a different perception of contact. So that has a manual error to it. That adds a manual error to our TLC plate. So that was reduced by using the spraying technique. So our samples are sprayed to HPTLC plates, and we get this results. So as the band application is evolved, so the resolution is better than in spot application, as well as we can separate more samples and more standards as the resolution is better, and you get to know more separation to your samples. So this is what the new HPTLC is. Then again, all the pharma company usually use a 20 by 20 plate and develop it to 135 centimeter, 135 millimeter. But in HPTLC, what we do, we use a smaller plate than that, half of the plate, that is 20 by 10 plate, and we developed till 62 mm. It is officially in pharmacopoeia, so we can use this plates for the pharma application. Here what happened, when we use HPTLC plate of 20 by 10, more samples are there. So this is a miniaturization of your TLC plates, which will require more solvent to work on in TLC plate. So less solvent is required in HPTLC plate, less time is involved. We get more sharper bends, and more samples are done in the same plate. So that's why we are practically using HPTLC 20 by 10 plate, which covers half of the development and which requires half of the solvent. Now, what are the regulatory acceptance? So I am only telling that it is regulatory accepted, it is regulatory accepted. So these are the things which are accepted. So these are the acceptance of HPTLC. It is a USB chapter 203, which was officially from 2018. HPTLC methodology and fingerprinting identification of materials of botanical origin. Its name is under this. We have USB chapter 621, which is scanning densitometry for quantification. USB 202, which has a identification of fixed oil. We also have USB chapter 2251, which is synthetic API in herbal, which is specially used for HPTLC MS. We also have USB chapter 1064, which is parameter selection chapter. In this chapter, we have studied all the parameters. They have given all the parameters, how you have to choose your plate, how you have to mark it, then how you can keep the distance between the track, what track distance should be there. So all the parameters which are usually done in HPTLC are explained in this parameter. So this is a very important USB chapter, that is 1064. Then EP chapter 2.8.25 is very identical to the chapter of 203. We also have IP, Chinese pharmacopoeia, and WHO chapters coming in the coming year. Then these are the compliance highlight. So these are usually the compliant. So we all the pharma company has USB FDA 21 CFR rule 11 compliant. So this rule is simple. Whatever you do in your instrumentation, the software should generate the report first. Then all the things you do, that is you enter any values or you do any changes in your file, it should be entered in in the software. So that thing is followed by our software, that is it is fully regulated 21 CFR rule compliant. So this rule is GLP and GMP compliant. So every QC, R&D and manufacturing unit has to follow USB compliant 21 CFR rule. And in this rule only it tells you that IQ/OQ should be done by each unit or each module of the software. So all the module of the software we do IQ/OQ. All data are encrypted and password protected database. We have digital signature till 5 level. So if you want to check reports in 5 level, that is from QC manager to assistant manager to the MD, so you can check and you can have a digital signature of them. User access control up to 7 levels. Full audit trials for analysis and system. So all the users and instrument data is stored in software. Built-in backup tool with scheduling. So this backup tool is very important nowadays for IT purpose and for pharma companies, because everything you store or every batch study you do has to store in a backup tool. So this backup tool, you can do an automatic scheduling also. If you forgot to take in Friday, then Monday it will beep a message like an alarm that you have forgot to do your backup on Friday. So please take it today. So like this, scheduling option is also there. Then these are some highlights of our software. So user rights level management. So here you can give user right, like different persons right, and they can do credit or do analysis, set read on flag, create and edit methods, diagnostic tests, system settings, licensing, user management. So these are the user rights which you can give easily to the person. Then you can give user ID length, password length, then allow the user to reuse the password, then how much chances you have to give, so that the if he enters 5 times or 3 times wrong password, then you can change the password, or you can change your password every 90 days. So this all privacy system, user management system is there in our Vision Cat software. Then regulatory settings. So this I was telling about the e-signature. So you can give e-signature settings to all your company members. Then system logger. So here we have who, here we note that who has logged in, what changes he has done. So all the things are logged in. Then we also have a built-in automatic backup tool. So this is the built-in automatic backup tool where you can select the drive where you have to do the backup. You can select the time and date, and then you can select the days. So number of backup also, it will give you 10 folders, 13 folders, 15 folders, and you can save the changes and then you can schedule your backups. So these are the features which are fully regulatory compliant. So now we will come to the application part where we have, when we will start our application. So first step is of sample preparation. So very easy to prepare your all the samples, pharma related sample. You have to weigh your sample, then add the suitable solvent to it, sonicate it, centrifuge it, and you can use the supernatant. When you are having tablets and all, the same thing you can do. You can crush the tablet, you can weigh your tablets, then add the suitable solvent, sonicate it for 10, 15, or whatever your monograph says, then centrifuge it for 10 to 15 minutes so that all the placebo and all are settled down and you only get the supernatant which is dissolved your solvent. Then this is the stepwise practice of your HPTLC. You first have to activate your software. You first have to make a method file. Then you have to make an analysis file. Then you apply the sample onto your plate. Chromatographic development you have to do. Image documentation, all the documentation part you have to do. Then you can go for densitometric scanning. Once you have scanned, you can get the reports, or if your substances or any sample which is not visible to you in 254 or 366 nanometer after development, then you can go for derivatization. Again, you can go for image documentation and scanning, or you can go for MS data, IR-NMR data, bioactivity, and effect directive analysis. So this all thing you can get through a single plate. So this is the power of HPTLC. What we say that is through a single plate you can get many data. Like you can derivatize the plate, again you can confirm it, double confirm it actually, and then you can again confirm it with MS data. So this is again the triple confirmation test. Then you can study the effect directive analysis of that bioactivity analysis you can do in the same plate. You can also go for isolation study and go for IR and MR. So this is the R&D part which you can do, and it is very important for a pharma R&D purpose. Then this is the chapter 1064, which is very important. So that's why I'm going to explain it to you, because it is the basic how you can apply your samples and what is the special recognition of a spot sample and a band sample, how you can convert your spot sample to a band sample. So here, first of all, I will explain you about the spot versus band. So this is the thing which CAMAC has tested. So they have tested the spot analysis. So here you can see, after certain period of development, it gets bulges and merge into one another. But here, when they have applied band type, it doesn't get merged, it gets resolved, and after certain development also it doesn't get merged. So it is best suited to use a band application than the spot application. Then not only that, but they have studied that what length of band you should use so that you can get a better resolution. So here you can see 2 mm band, 3 mm band, 6 mm band, 8 mm band, and 10 mm band. Of that, they have found out that 8 mm band is the best suited for your sample analysis. Then distance from the solvent. So what distance substances should travel so that it can get best resolution. So that distance also they have traveled, and of that they have found out that 1/3 of the plate should be covered. So 7 centimeters is the best distance it should cover for a developing nesting. Then they also studied that different changes of chamber saturation and chamber configuration also they do a best result. So saturated, unsaturated preconditioning they have done in twin trough chamber, they have done in horizontal development chamber. Of that, they have found out that twin trough chamber with saturation, 20-minute saturation will give you the best results, best resolution. So they have confirmed in this chapter that you should saturate your twin trough chamber for 20 minutes, or whatever the monograph says. Sometimes the monograph says it should be saturated till 1 hour. So you can change it to 1 hour. But if you don't have any monograph, then you can just follow this USB chapter and you can get the results, reproducible results. Then they have studied relative humidity. So same sample has been studied in different relative humidity, and they have found out of this. They have found out like 33% will give you the best results, other than 5%, 45%, and 75%. So you can always do your samples in 33% relative humidity. Then application pattern. So application pattern should look like this. This is a 20 by 10 plate. You have to write your project number or any number, specific number which you are doing. Then the distance between the sideways to your first band should be 20 mm, because if you are uh, doing application in the sideways, then you might there might be a chances of getting a edge effect. So for that, for removal of that edge effect, 20 mm distance should be spared so that you don't get that edge effect. Then B distance, that is from one band to the midpoint of another band, it should be 10 mm distance so that it doesn't get merged into one another. Then the C distance, it should be 8 mm. So this 8 mm distance is the single band distance. Then the D, that is from downwards to the bands. So this distance you should keep as 8 mm, because when you keep your plate in a twin trough chamber, so that band should not get dissolved in your solvent. It should develop with your solvent. So that's why you have to keep 8 mm distance from downwards. Then 62 mm. So E is 62, that is the developing distance. So according to the USB, the sample should cover 1/3 of the plate, that is 70 mm distance it should cover. So this is the basic pattern, application pattern, which you have to always follow for getting a reproducible results. Then ChemAg has also done a certain advantage for QC and research lab. So certain features are as follows. So first feature is it is an HPTLC instrument comprises of independent instruments and controlled by one software. So it is an independent instrument. It is not like HPLC or GC where you have the instrument attached to one another. Only one person can do your sample and standards at a time. But here in HPTLC instrumentation, there are different modules for different purpose. So this is a modular system which comprises to a very much HPTLC instrumentation. So up to 6 or 7 analysts at a time simultaneously can do their different 6 analysis at a time. So one analyst can do work on application, one analyst can work on development, one analyst can work on derivatization. Likewise, simultaneously you can operate 6 to 7 analysis at a time, which is easily upgradable. That likewise, this software is followed everywhere, and if you take one module, that is visualizer and applicator, and if you add after certain year, if you add scanner, so that software is compatible with scanner also, so it gets easily upgradable. Then the database oriented software or QC. So for QC purpose, what things are required, that it should track easy of your samples and standard files. So you just have to type your name onto the Explorer, and then it will easily track your file name. You can compare, generate reports, multi-security of data. Each analyst is given its password protected. So whatever that analyst do is recorded in the software. So if he does changes, that is also recorded in the software. So that is the basic thing which is needed for a QC department. Minimum use of solvent. So this is the bestest advantage of HPTLC, that only for 15 to 20 sample it requires 30 ml of your mobile phase. So it is very economical and very little waste is produced. Analysis is demand, no preparation time is needed, no disturbance to other users, and you can report your sample analysis within 60 to 90 minutes. Trustworthy Anchrom support since 1978. So we are super specialist in HPTLC from last 40 years. We have assured lab in Mulund, which is situated especially for the customer queries and analysis purpose. So you can rely on us. And we also have a maintenance team, that is a service team of 14% who travels all over the world for customer satisfaction. Then Indian Pharma Company and HPTLC. ChemAg is the standard for HPTLC worldwide. It passes all the inspection. We have more than 100 pharma companies who are working in HPTLC instrumentation. They all have passed their inspection in India, in US FDA, WHO, Health Canada, MHRA, EU inspection. Full documentation is available, such as design qualification, CE certificates. All the certificates are given from the Switzerland. Then we have software and hardware which are specially made in Switzerland. That is ChemAg. Then these are the sample applicator which we usually use for applications. So first is Linomat 5. This is the semi-automated one. So every time you have to remove your syringe, you have to rinse it, then keep your sample, then take your sample and keep the syringe again in the turret, and then press enter. So it will apply automatically. Only the rinsing and the filling is semi-automated, that is manual. So this is the semi-automated Linomat 5. This is fully automated. You have to keep your vials filled, standard and sample vials. Then you have to give command from the software that this much ml of my standard should be spread, this much ml of my sample should be spread. It will take, it will rinse the syringe. These two bottles are there. One is for rinsing solvent, one is for waste. It will automatically rinse the syringe. Then it will take the sample and it will automatically apply. After application, it will tell you that application is complete and you can go for next module. So this is fully automated ATS4. Then we have Applicator Pro. This is also fully automated. Only difference is there, it is more precise. That is, it is containing 10 microliter syringe. Here we have 100 microliter syringe, 25 microliter syringe, and 10 microliter syringe also, but it is not that much use. But here we have only 10 microliter syringe. It is very precise, and the sample quantity tray, that is sample tray, is 75 for 15 for 5 plates. So all these things are very precise. Regulatory norms are used and integrated to HPTLC system. So always we have a band application. Band which are spray on, which work on spray on technique. It is very precise and accurate, and it requires GLP 21 CFR rule, and we get a traceable data. It is also integrated to system managers. We can also do a stability check study on our samples. So here we can spot the samples, then we can develop it in one mobile phase. Again, we can develop it in the same mobile phase or second mobile phase. So this is a 2D check of the chromatogram. If your compound is stable, it will show you an angle type like this. But if your compound is not stable, it will show you many compounds and it will not show you an angle type inclination. So here you can study on the same mobile phase 2 times you can develop, or you can change the mobile phase first time and second time. So this study is also known as stability check of our samples on silica gel layers. Then these are some advantages of HPTLC. HPTLC is very simple, reliable, safe, fast and safe technique. Why it is a simple and fastest technique? Because only within 1.5 hour you can get your sample results, and that samples also not 1 or 2 but 15 sample results at a time. So per sample you require only 2 or 3 ml of your solvent. Then ultra low running cost and maintenance. So 2 ml of your solvent is required as a mobile phase, and maintenance also you require only for the syringe rinsing purpose. That is also 1 or 2 ml for rinsing purpose. Then multiple detector eyes. So you have a tungsten lamp, you have mercury lamp, you have deuterium lamp. So this is a multiple detector eye system. You don't have to attach your lamp from outside. As in HPLC you have to attach your mercury lamp, again mercury FID lamp if you are using mercury 366 nanometer. But here you don't have to attach any lamp, extra lamp. Here you have 3 lamps already given, and it covers 190 to 900 nanometers. It has electronics and bioactivity also you can do. You get a huge data from the sample, UV range, visible range, fluorescence range. Also you can do a spectra study, purity study. Then you can derivatize your sample. You can again double check it. Again you can go for MS, you can check it. Then you can also do a bioactivity study. So through a single plate of 15 samples, you get this much data. So huge data is generated from a single plate. Then we have a chromatographic ISO development system. So these are all the models which are mostly used for development purpose. So first is the twin trough chamber. So this has a twin trough. So this trough has an advantage of giving 45-degree angle to it, to your plate. So whenever you develop the plate, you have 45-degree inclined to your plate. But when you use a flat bottom chamber, it doesn't gives you 45-degree as accurate as in twin trough chamber. Then the solvent which you use, that is 20 ml, is segregated between this trough as commonly, as equally, that is 10 in this trough and 10 in this trough, which is used for development. 10 ml is used for saturation, 10 ml is used for development purpose. So you require very less amount of your mobile phase. So these are some of the advantage of trough chamber. But as it is a manual development chamber, we cannot control the humidity. So for controlling the humidity, we have ADC2, that is automatic development chamber, where you develop your plate automatically. The sensor sense how much it is developed. Once it is developed, it will automatically take your plate up. It will dry your plate automatically. Here we have one more chamber, that is a humidity chamber, where we keep a saturated salt. So different saturated salt give you different types of humidity. Like KCl saturated salt will give you, that is potassium chloride saturated salt will give you 47% of humidity. Then sodium chloride will give you 75% of humidity. Molecular slaves will give you 5% humidity, and potassium acetate or magnesium chloride will give you 33% of your humidity. So here we can control the humidity, and it is an automatic development, which is according to the USB. Then again, we have gradient type of development, where you can use different type of solvent. So not only one time, but at least 7 to 8 times you can change the composition of this gradient system. So if you have a plate, you have to develop 10 through one mobile phase, which is their ABC. It is 100, 20, 30. So you can do 10 development till this composition. Then you can change the composition to 40, 60, 60. So like this type of composition you can change through this gradient type of a system, that is AMD2, automatic development, automated multiple development system. Then we have robotic development pro module, which is very similar to the ADC2, but here development happens very fast. So the chamber is designed such that through the gas phase only the development happens, and every time you don't have to put a mobile phase. So the mobile phase is circulated into that chamber, and the gas phase is also circulated in that chamber. So this is the development module pro. Then again ADC2, which is a USB compliant technique, as it controls humidity. It is controlled by software, fully automated. It gives you humidity 33%. It gives you layer equilibration. It gives you automatic monitoring of your software, solvent front, automatic drying of the plate, and it is integrated to the HPTLC system. Robotic Pro is also having these features. Then gradient system, you have 5 solvent option where you can do n number of steps. For examples, for example, like suppression of lipids, phospholipids, ceramide, cholesterol, fatty acids are done by AMD2. Then this is the type of development which we do. Then certain points are there for the mobile phase in HPTLC. Very little consumption is there. 30 ml of your mobile phase is needed for 15 sample. Any composition of stationary phase and mobile phase is possible in HPTLC. No preparation time, no degassing time is needed, as only little amount of solvent is consumed. Then gas phase in HPTLC is mostly 20 minutes of chamber saturation is essential, and 33% of relative humidity is needed. That is by using saturated solution magnesium chloride salt. Then we have stationary phase in HPTLC. Mostly we use 60F 254 silica gel plates, but you can also use RP plate for certain experiment. We also have HPTLC pre-coated 20 by 10 glass plates, which are used for pharma applications or for pharma companies. Then next module, visualizer for photo documentation. So one of the advantage of HPTLC instrumentation is that whatever you do to your plate, or whatever you do experiments to your plate, you can see. So this is the basic advantage of HPTLC. So it is a visual technique. No other chromatographic technique is a visual technique. Only thing you can see is after it goes to detector, or anything you can see is in the detection point. But here, before detection, you can keep your plate in visualizer and you can see what you are getting, and then afterwards you can go for detector. So in visualizer we can take photo in white light 254 and 366, which is a major concern lights. Then you can also take photos before and after derivatization. So here is an example of that. Then you can also stack your data, stack your samples, and you can compare it like you do batch to batch study or stability check. So here is an example of comparison of multiple curcuma samples from different plates. Not only from different plates, but different samples also you can do, different species also you can do, different days you can do, different labs, different analysis. So all data are traced, and you can just tag the data and you can compare it in the same file. Then next module is the quantitative evaluation scanning densitometric. So UV visible fluorescence range. It has 3 types of lamp: deuterium lamp for UV region, tungsten lamp for visible region, and fluorescent for fluorescent it has a mercury lamp. So for whole data, whole wavelength range is covered, that is 190 to 900 range. You get a fully automatic spectra and scan. It is very sensitive and gives you a reproducible results. No, so not only chromatogram, not only RF value, but you can compare your spectra data also through the scanner. Then you can plot the calibration curve. You can have a 3D display also. So this is the schematic diagram which takes place when we click on the button to scan. So first one, we select the lamp. Then the lamp light passes through the monochromator. It reflects through the mirror. Then one slit, we have to select the slit. So through the slit, the light passes, means the light is filtered through the slit, and depending upon macro and micro, that is TLC or HPTLC lens system, it passes through the beam splitter and photomultiplier tube. Then it goes to the plate and it is detected. So this only takes place within 2 minutes. Then through the chromatographic scanning, you get the absorption spectra, that is the RF value. You get the fluorescent scanning, you get the UV visible spectrum, and the calibration curve, that is the linearity curve. Then this is the spectral comparison. Again, the example of spectral comparison, if you are getting same RF values, but if you are getting different spectrum bands, so then you can double confirm that the sample and the standard which you are seeing is not same. Then not only for the identification purpose the spectral value is taken, but for the purity band also, purity purpose also you can do. That is, if you are doing any isolation study of any unknown impurity or known impurity, then you can change whether the impurity is pure or not. So for that purity check, you require the spectrum. Then this is the example of what you can do through the scanning and HPTLC instrumentation. So this is a quantification of tetracycline, an antibiotic compound. So what she has done, what the student has done is she has injected the tetracycline, that is antibiotic antibiotic standard, tetracycline standard, to the ground plant, and she has studied how much it is absorbing in root and shoot by HPTLC. So you can quantify it. Then not only that, other example is of amoxicillin. So the same thing she has done, she has injected in the plant, and she has found out, she has quantified how much nanogram the plant has absorbed from the soil. So this is example of that. Then not only that, you can quantify through the Apexa APIs such as Apexa Beam. So you can quantify the tablets, you can get the linearity curve, you can get the CV value, R square value, slope value, sorry, slope values. Then you can also quantify and do the assay values at the specific wavelength. Here we have done at 280. So likewise, you can do at different wavelength according to the lambda max value. Not only that, you can do the impurity profiling of the compound. So here is an example of carbazole samples. So here you can see these impurity profiling, impurities which are coming in the main compound. So that also you can find out through the spectra. So here you can find out the spectra of unknown matters is at 291, 248 and 251. Then not only that, you can do multi-wavelength scanning. So through a single scan, you can do different wavelength scanning. Here is, for example, we have done at 268, 205 and 311. So here is an example of multi-wavelength scanning. So this is a simultaneous estimation of 3 APIs. So not only for the simultaneous estimation, but also through the lambda max value, you can estimate in that particular API for that particular sample. So here you can see the spectra values is coming different for this 3 API. So you can either scan it at that particular lambda max value, or you can take one single lambda max which is overlapping lambda max of that 3 APIs. So what we have done is, we have taken this is a simultaneous estimation. So in through one plate we have done overspotting of these 3 standards, and then we have found out the spectra of these 3 standards. Then it is found out that in 240 nanometers all these 3 API overlap with at a certain point, and that point is taken for further estimation or for further calibration points. So likewise, we have done this 3 simultaneous determination of these APIs. Then we come to other module, that is the automatic derivatizer. So piezoelectric, it works on piezoelectric fine spraying. For the reproducible results you get, 3 ml of your reagent is required for 20 by 10 plate. Automatic reagent vapors are removed. No fume hood is required, and it works on green technology. So post-chromatographic derivatization is mostly unique to HPTLC, as we have to spray our reagent to the plate. So you just have to spray your reagent and heat the plate. You get the additional information about the sample. Also the city of the functional group and class of compound also you get. You get ultra high sensitivity and visual confirmation. So this is a type of results. This is usually used in herbals, but we can also use for APIs. Likewise, further example we have given. This is example of a artimeter and dihydroartimeter, which is mostly used for malaria tablets. So these two compounds are usually separated when 10% sulfuric acid is sprayed to the plate. So this mostly it is quantified after derivatization with 10% sulfuric acid. So this is example of how you can use HPTLC for derivatization purpose, and how you can estimate a compound, as well as you can do a specificity study in the placebo as well as tablets. Then again, this is example of meltafosine, which is used for skin diseases in South Africa. So in this also 10% sulfuric is used for estimation or for visualization. So it was best suited by HPTLC than by any other instrumentation, where you have to prepare your samples with many things so that it gets visible, and then after that you cannot use the colored things to your HPLC column. Whereas here you can derivatize the plate and you can easily see the product onto the plates. So this is example of meltafosine tablet after derivatization with 10% sulfuric acid. Then we have HPTLC MS interface. So it is used for confirmation and characterization, chemical synthesis, phytopharma, pharma R&D, food, organic study. Then it is mostly suited with MS interface or HRMS, MS for selected fractions only. This is the schematic of how TLCMS is coupled with the MS. So an HPLC pump is used to deliver the solvent to the pump, to the interface, and through the inlet of the interface the solvent is pumped. Then it will go to your plate. It will elute your bands, and then with the help of the solvent and that band, the eluted particles get dissolved in the solvent, and it will go directly to the mass spectrometer, and it will give you mass to charge ratio. Then Shimanzu and Anchrom joint poster for ASMS conference at USA was done. This is the USB chapter which is used for 2251 for HPTLC-MS. So not only that, but not only known impurity, but unknown impurity also you can find out through HPTLC-MS. So here is an example of finding out known and unknown impurity. So they have found out, they have eluted with HPTLC-MS, and they have found out the mass to charge ratio. Then this is the analysis of insulin samples by HPTLC-MS. So different species of different insulin was done with the standards and samples. So the same reference spectra and human insulin spectra was compared, and it was found out that it matches. So this was the insulin which was taken by the humans. So like this type of things you can do with HPTLC-MS. Then the advantage of HPTLC-MS hyphenation is it is very simple to use. Only 3 minutes is required. Only pure spectra is taken and eluted. Cost effective MS analysis can be done. You can do 100 mass per day for the selected fractions of interest. It increases the LCMS productivity. So this is the added advantage to LCMS, because we don't have to use columns. We don't have to pass the solvent or samples to the columns, which the columns are very heavily used. And for that purpose, we are hyphenating to HPTLC MS. Then this is the full system data output. So we have applicator through which we apply your sample, then we develop it, we take the photo documentation, we see the photos. Then afterwards, once we have confirmed in visualizer, we go for scanning. We get the chromatogram, we get the linearity curve, then we go for MS hyphenation. So here are the MS hyphenation data. Then you can double confirm it with derivatization technique. You can get the visual technique, the visual double confirmation. Then these all modules are handled through a vision cat software. So this is the whole new concept of bioactivity, CAMAC bioluminizer. So it is mostly used for toxicity study, which is done for mostly API in clinical trials. So this is example of toxic study for wastewater. So you can see this is the results which we are getting when we are applying our wastewater sample. So we are detecting with albovibofishery bacteria, and this bacteria is very sensitive to the toxic effect. So it dies onto the plate and gives you blackish color. So all these blackish color bands which you see is the toxic compounds of that wastewater sample. Then bioluminizer application is for selective detection of biological activities in synthetic mixture, natural product extract, toxic profiling of water, toxic residual material detection, food beverages diet treatment, screening of toxicity, toxicity residual in soil. So this is example of bioactivity product in marine sponges. So what we have done, we have applied it, gradient development, then we have taken photo. So ABC is the photo through the visualizer. Then we have derivatized it or use the bioluminizer for taking photo. And we have found out through TLCMS, we have found out this toxic compound, that is the black color compound, what it is through the MS. So this is example. Then you can also use for drug discovery. So effect directive analysis is also used for drug discovery. So here is an example of that. So this is ABC. This is how plate will look like when you have sprayed with VSR. Then after VSR, they have studied with B substatus and acetyl in C. So all the bands which are coming is then checked with HPTLC-MS hyphenation for their mass to charge ratio. HPTLC method development validation, it is very simple, takes only few plates and 1 day to develop the method. For validation, it takes 3 to 5 days. All the whole cost of validation is 25 to 50,000. It is possible to create and validate 10 to 15 methods per month. HPTLC validation is followed by ICH Q2 R1 guideline. In this guideline, all these steps can be done by HPTLC, that is specificity study, precision, both interday and intraday, accuracy, linearity, limit of detection, limit of quantification, and ruggedness study. These all parameters can be done by HPTLC instrumentation. So here is an example of our validation study. So quantification and specificity study for glimepiride tablets in Glimemer 1 mg tablet. So here what they have done, they have applied sample, blank sample and standards, and also they have applied mobile phase. So here in this plate you can see that in blank sample and mobile phase you cannot see any band. So this means in solvent, mobile phase and blank sample, all the bands are absent which are present in sample and standard. So this method is specific, or the impurity or the blank samples is not there, not showing any bands in the given RF value. So like this type of study you can do. You can quantify the samples. So here you have quantified the sample. It gives you CV, coefficient variation, and R square value. It has a linearity curve, which is a 5-point linearity curve, and 3 uh, replicant. So you get the assay value. From this assay value, you can determine how much mg was present in your tablet and how much mg you have gained from the experimental value. Then you can compare the percentage, and if it is 98 to 102%, then you can go for further study like reproducibility, recovery study, like this study. So here one recovery study is given. So this is a mixture, this is the standard. First we have applied sample, then we have applied standard and sample mixture. Then again we have applied standards from 80%, 100%, and 120%. Then we through the software only we can generate an SV Excel sheet, and through that Excel sheet we can copy paste the area to the Excel sheet. We can get the average value, we can get the expected and percentage value, and then we can find out the mean percentage, and we can find out the recovery, how much you are getting. Then not only that, but the forced degradation study also you can do. You can all these things are done for the clinical data or method validation study. So for forced degradation in sodium chloride, that is in basic, acidic, in for phytolytic, thermal, and in acidic medium. So all these medium you can test, and as it is a visual technique, you can visually see and generate your data. Like this, in sodium chloride it is not getting degrade that much, but 2 degradation is there, as well as in H2O2 it is getting 1 degraded compound. Then this is the standard. Then again in phytolytic it is not getting degraded, whereas in thermal it is getting degraded, and you can find out the percentage of how much it is getting degraded. In HCl also it is not getting degraded. So like this, visually you can find out the degraded study. Then books and literature are available on HPTLC. So you have a ChemAg site where you can find out the application note. So different types of application note you can find out. You have HPTLC association, you have Ayurvedic pharmacopoeia book, then we have HPTLC book on medicinal plants, quality standard plants, plant growth analysis book. We have HPTLC book on food analysis. Then we also have a quantitative analysis of pharmaceutical formulation book, which is written by PD Sethi. All the 3 volumes are used for HPTLC analysis. Then there are different application area of HPTLC like pharma, food, botanical, industry, engineering, chemicals, mixtures, and cosmetic. Then in food adulteration, so for production or for manufacturing of these APIs, oils are needed. So you can also find out what oil is adulterated. So you can, this is example of adulteration of oil, where through GC it was done, but through GC it was not found that it was adulterated. But when done with HPTLC, it was found out that one adulteration product is there and it was quantified. Then you can also find out the visual thing versus false detection. Likewise, here is an example of that. You have sample and standard. So standard is showing the same RF value as of samples in the sample. So what you will do is you will see that it is present. But if you will compare the spectra of standard and sample, it is not coming, it is not matching. The RF value is 0.22, but it is not matching your spectra value. So it is find out through the spectral confirmation that whether it is if it is matching with the RF value, but it is not matching your spectra, then you can easily say that it is absent in your compound. And also you can find out through the visual confirmation test, that is derivatization. So here you can find out that it is coming different color. So you can easily tell that I had double confirmed that, and the standard is not matching with my spectra. So this is the example. This is one example how you can do a double confirmation test of your sample. Then the other example or other field of pharma sector where HPTLC and HPTLC MS can be used, that is API manufacturing for clinical API batches. So in these batches, what they can do, they can compare the batches of different process. So here, for example, they have taken pure solvent, then they have taken pure solvent with other solvent. Like this, they can do different manufacturing processes, and they can compare it through the visualizer. Then not only that, for the identification of amino acids, so amino acids is one of the synthetic peptides for pharma products. So there are two types, natural and non-natural. So you can find out the natural and non-natural through the HPTLC instrumentation. Then the degradation profiling of antibiotics. So different degradation profiling you can do, as I have told earlier. Cleaning validation. So one of the advantage of HPTLC in pharma sector is of cleaning validation. So in cleaning validation, this what happen is many manufacturing companies are there. So what they do, they pass a solvent and clean their instrumentation before passing the other batch compounds or other samples. So what through this all the through the solvent is passes to different lines. So these lines are opened and the solvent is extracted, and they can be checked in HPLC or other instrumentation for the cleaning purpose. So if any impurity is there, then they have to clean again. So for that, HPTLC is mostly you can use HPTLC because at a time 15 to 20 samples you can check, as well as it will save you time and cost as compared to other instrumentation. So cleaning validation is one of the most advantage application of HPTLC. Then again, content uniformity check. So you can check the content uniformity also through HPLC also, through GC also, but there you will take more time because the content uniformity, in content uniformity, you have to check the content of your sample, which is which will be very large. So the concentration will be very large. So for washing and all, you will require time. But here, there is no waiting for the run completion, no waiting for the other batches. You can put 10 to 20 batches in a single plate. You can compare it and you can easily pass the batch. You can just have to put 100%, 120% and 80% standard, and then rest all samples you can put. For example, here they have shown 60 samples. They have used both side of the plate for application, and they have developed it with two sides. Then not only that, but for clinical trials or pharmacokinetic study in plasma samples, also you can use HPTLC. So this is example of that. Then not on till now I had explained you all the manufacturing processes, examples, applications, all the R&D applications. But now HPTLC will be using for quality control purpose also. So for that, HPTLC Pro has been launched, and this is the video.
As world leader in planar chromatography, CAMAG strives for innovation. We constantly shape the future of analytical solutions. HPTLC distinguishes itself by its strength in the visual and comprehensive evaluation of complex samples. But how can we combine this valuable aspect with speed, reliability, and cost efficiency? Introducing HPTLC Pro, our newest contribution to improve analytics, fully automated, reproducible, to the point, making hyperspectral evaluation of analyte mixtures a reality. HPTLC Pro, leading high-performance thin-layer chromatography into the next dimension.
So 5 modules will be launching. Till now 2 modules has been launched and more 3 has to be launched in the next coming 3 years. And this is the working of a HPTLC Pro unit in our lab.
We will probably change to after, not now.
So the last track on the plate has been applied, track number 15.
Yeah.
Now the needle has gone for rinsing. The plate is now finished. You can see the plate is coming out. It is moved to the transport automatically. And now it will be fed into the developing chamber.
It's asking for the second plate.
And the second plate is also going in. So you can see the automation. We are doing 2 analyses back to back. So the second plate will now start being applied while the first plate is being developed.
So the basic concept of HPTLC will not change. You can do simultaneous experiments, that is 5 plates you in all the 5 modules, and 6th plate will be waiting for the application. So like this simultaneous estimation you can do, but this unit will be automated one, which will be specially designed for QC purpose, that is quality control purpose, where they have fixed method. So they in the start they have to start the method, and it will automatically go to the applicator. It will do the application, then it will go to the development. So all the thing will be done automatically, which nowadays we have to do manual, or we have to take the plate. So there, according to the QC department, what happens is the manual error is more, but here now the manual error will be subtracted because it is an automated system and closed box.
So there is a fresh plate being applied now, and our older plate being developed in this chamber. So that's the automation.
So this will be the automation, that is the next generation Pro. Now, not only we give the application support, but we also support all the students, all the members which are doing HPTLC R&D, or which are publishing papers. So here is an example of that. We have supported Dr. Vandana Gawande ma'am for the Berlin Symposium of HPTLC Symposium, and she has won Most Promising Researcher Award for her paper. Then in 2018 at Bangkok Symposium, we have supported Indian students of Yugendra Patil Madam, Dr. Shunita Shailajan from Ruya College, and they have won the gold prize for their poster. So Anchrom philosophy is we are technologists, not traders. So Anchrom will be your analytical partner. It will not only supply you the instrument, but keep your instrument running due to its excellent service, laboratory, and customer focused support. So this is our newly expanded application lab. It is having unlimited customers to the support. HPTLC will be accessible to all the Indians who wishes to do HPTLC papers or publication. All ChemX software instrumentation for training are available, and for analysis are also available. We have 14 analysts, including 2 PhDs. We have separate sets of instrumentation for hands-on purpose. We have PhD earn and learn scheme. Lab is always available for publication work. We give free trainings at Anchrom for all government employees. Training duration is flexible. We have mutual convenience date. We certificates are issued at our lab on-site and off-site. We have trained 5,000 scientists. MOUs with certain universities has been signed. So our email ID is hptlc@anchrom.in, and this is our WhatsApp number through which you can contact us. We have an Anchrom YouTube channel. You can subscribe us for latest videos and this thing. So Anchrom's aim is to make India the world largest to be used of HPTLC, and our Anchrom lab gives access to all the Indians who wishes to use HPTLC for their publishing work purpose. So thank you from my side. Now Now I will be showing you certain case studies. So let me share my screen once again. And so 2-3 case studies are there. We will just see it. So first is impurity profiling. So this is the sample which is used. This is the data which we get once we have generated the data. We get the analysis file. We get who has created it, who has modified it. Then we get the chromatogram, plate layout, every step data we get in our results. We get the plate image. We get the, so this is the example of impurity profiling. So what we have done is, this is the mean substances and this is the impurity in which we are testing. So what we have done, we have uh, attended the limit, that is the limit is 0.125 in this sample, in this mean substances. So what we have done, we have applied the limited sample and we have just compared it, whether if it will be present, then it will show you this band here, a bluish color band. So this is just an example of impurity profiling, how you can interpret the data of impurity profiling. So you can find out, you can see the peak here of that impurity, whether it is absent here. So this is example of an impurity profiling. Again, one more example of impurity profiling. So here you can find out through this visualization technique only, you can find out that the substance is not showing any impurity. And if you dilute the substances also, you can see that it is not showing any impurity. So this is the data how we get. Then again, limit test. So how we test the limit, you will see the direct the results. So this is the type of limit test we get. So if it passes the limit, it will come as a tick mark. If it is not under the, if it is absent, it will show you an A. If it is present, it will show you red color. The test fail, it will show you red color. Test passes, it will show you green color. So all these are passing the test, so it is showing green color test. So this is the limit test example. Then validation example. So this is the linearity. So you can see this is a two standard which we have plotted the linearity. These are the samples. And we have the linearity curve here for lupeol. We have the assay values. So this is beta cetosterol, CV value, R square value. Again, if you are doing reproducibility, so it will be giving you the results of reproducibility. So it will give you the CV value and average value of that, and it will give you the deviation. And if the CV value is less than 2%, that's mean it is passing your reproducibility test. So the same thing is shown here. Then specificity study. So through the photo only, through the visualizer photo only, you can find out that it is passing your specificity study. See, in mobile phase and in solvent, it is absent. The sample and standard are absent. So you can easily find out through the visualizer the specificity study passes. So this was the certain case study. So now I have completed this webinar. So thank you for the patience and listening me so keenly to all the participants. Hope I have enhanced your knowledge with my expertise, and hope to see you all on 6th April, the next webinar series that is of forensic science application. So thank you from my side.
Yeah.
Over
Thank
to
you,
you,
everyone.
Sekat sir and Ankita.
Yeah, thank you, Sneha, for briefing us with this, and hope all our participants have benefited. Have we taken up any questions up till now, Sneha? No.
Sir, no.
Okay, okay. So we'll quickly go through the questions which have been dropped in the chat and the question answer session. And if you have in the meantime any more questions, just raise your hand. We'll be unmuting you and we'll be directly answering your questions live. So the first question which I see in the Q&A box is, I'm using HPTLC for herbal analysis, but sample preparation only takes time. We are using plant drug analysis as a reference, how to reduce the sample preparation time. Now, this is very interesting because sample preparation is an important aspect for any chromatography. There are certain limitations to the grades of chromatography instrument you are using, like HPLC, GC, HPTLC, and all those things. And sample preparation is very key aspect. So in HPTLC, this problem is very simplified because the tolerance limit of matrix in HPTLC is much more higher. You can load a sample with a higher matrix load onto HPTLC, those plates which are there, the stationary phase, and analyze the sample, because when you are selecting a particular mobile phase, only your selective analyte comes. The disposable nature of the plates allows you to use it without any concern of matrix effect. So sample cleanup is not required that much. For herbal, normally methanol, the USP guideline states a very simplistic procedure: 100 mg of sample in 10 ml of 1 ml of methanol, and you can sonicate, centrifuge, and use a supernatant for your sample preparation. If it is an extract, you take 10 mg in 1 ml, and that is how you can prepare. In case of API, when we talk about the pharma industry, the tablet itself, excipient, has a lot of weightage. You have tablet excipient materials which are there, the coating material are there. They really affect your separation, and that is why in sample preparation of tablets, you can directly take a tablet, dissolve in your solvent, centrifuge it, take the supernatant, and do the analysis. If the tablet has sustained release, you have to go for options of understanding what kind of a tablet property it has, whether it is a sustained release tablet or it is an immediate release tablet, because that is how the excipients are added, and that will give you the extraction technique has to be optimized along that. Whether it is immediate, it will immediately give you that much of quantity of the drug. If it is sustained release, it will take time for the releasing, and then you have to really crush the tablet nicely, homogenize it, and then extract it. So this is a very important step. Try to avoid the coating material by simply peeling it off from the tablet when you are analyzing any tablets with coating given to it. So such kind of an approach can be given to all these kind of analysis. For dissolution assays, we had done one experiment for miltefosine, which has been shown by my junior colleague Miss Sneha, and you must have seen that is directly from a dissolution apparatus. So we directly take the sample without any processing, we apply it onto the plate, and we develop it. We don't need to neutralize or things like that, and it can be directly applied onto the plate, and separation can be observed. So rest all procedures are very simplified, and as I told you, high matrix capability allows you to have a lot more matrix, because with your mobile phase you are selective in observing components, and that takes care of the sample preparation. Now when we talk about drug bioanalysis. Now, biomolecules are heavyweight molecules. They can be secondary metabolites obtained from organisms or plants, or they can be protein itself, enzymes itself as biomolecules. So the definition of being a biomolecule is very huge. So here, if it is a smaller molecule along, say, amino acids still, we can do the analysis smoothly on silica grade plates. There is no issue. If you have proteins to be analyzed, then there are special proteomic plates which are available from the Merck and other vendors, and you can utilize them from protein analysis. But if you have very high molecules, I doubt whether HPTLC will be suitable for that, because The plate has its capability, the stationary phase has its capability. It cannot handle a molecule of higher kilodalton weight size, and it will not be able to resolve. So I request you to check the biomolecules kilodalton uh, size, and according to that select the stationary phase. Normal secondary metabolites, sugars, basic sugars, monosaccharides, polysaccharides, they are quite possible. But if you talk about proteins and enzymes, they can be a little bit of challenging, and in that case you may either have to shift to reverse phase, or you may have to try with proteomics plates. But there are some limitations to that. In that case you may have to go for an alternative technique. Now, after that, there is a question is how to give pre-washing to the TLC plate. It's very simple. What we do is we add your methanol. Pre-washing can be given either with a organic solvent or a mobile phase. Organic solvent can range from methanol. Normally, most popularly, methanol is being used. You simply place the plate inside that, allow the plate to develop. You don't dip, you actually develop the plate. So what you're developing, the sample or analyte. Similarly, you have to develop the blank plate in methanol, allow it to run almost till the end of the plate, say 99 millimeters, that is 9.9 centimeters, if it is a 20 by 10 plate. And similarly, if it is a smaller plate or higher plate, if it is there 20 by 20, then you run it till 199 millimeters, that is 19.9 centimeters. Then take the plate out, heat it at 120 degrees Celsius for around 20 minutes, and then you can use this plate for your application. This process which we do, it is known as pre-washing, and I hope this answers your query. Then we have a question, can we use 20 by 10 HPTLC plates for automatic applicator? Absolutely yes. There is no problem for that. Our applicators are such that they accommodate the smallest plate size to the very biggest maximum plate size, which is 20 by 20, that is commercially available. Then we have what mobile phase is used for the separation of limonene. Limonene, if I'm not mistaken, is an essential oil. So ideally you can use toluene ethyl acetate in a ratio of 93:7. But there are also certain literatures which are available, and we will also check the reference paper, and if we find any reference paper, we'll surely share with you. But as of now, you can use toluene ethyl acetate. Then what type of a mobile phase can be used for diclofenac and aspirin? Yeah, this is something very good. So normally diclofenac and aspirin, we'll have to check the literature, but there is availability of, you can refer to your HPTLC analysis of pharmaceuticals by Dr. P.D. Sethi. There is a proper volume 4-volume book which is available, and you will find lot of combinations and drugs which have been worked upon over there, and mobile phases which are available, and you can pick and choose from that for your analysis. If we don't want any type of protein or fatty acid in the plate, what should we do? So normally we precipitate out, or by physical means we coagulate it by either chilling it, or you freeze it, and simply all the organic solvent will be available without any problem, and you can clearly separate it out. So that can be a case, or you can add some acid into your sample, but then you have to be careful that it does not degrade your analyte. So that can be, physical means are a better option, so you can look for that, centrifuge it, and if the molecule is acid stable, you can add some acid to the protein, so the protein absolutely precipitates out, and you can have the clear solution which can be analyzed. There will be more methods. You can refer to literature, thin layer chromatography by Joseph and Sharma. Then there are many books which give you practical knowledge of that. You can also refer to plant analysis of drugs by C.B. Clark is the author. You can refer to that book. That will also give you a lot of details. Natural dyes analysis, yes, we can use absolutely. In fact, for dyes, HPTLC is one of the best techniques you can use. So absolutely, natural dyes, we can do analysis with HPTLC. You will have a lot of reference papers which are available, and easily toluene ethyl acetate, because these are generally pigment compounds. So they work with flavonoid mobile phases. So you can just try with them, or you can try for toluene ethyl acetate based mobile phases. Add a little bit of methanol to increase the polarity, and you should get a separation of these dyes without any problem. If they, as I told you, if they are more polar, then you select a mobile phase like N-butanol, acetic acid, and water. This will also help you resolve many compounds which are highly polar in nature. How will you select slit width? So slit width is selected as per the size of the band. Normally it is 80% of the bandwidth, but in many a times if the conditions are not proper while developing, plates might go a little bit of tilted because of saturation issues or mobile phase composition issues. Then you can reduce the slit size, because in HPTLC all the samples and standard are on the same plate. So the whatever changes you make for a single track, that is being applied to all the tracks, so you don't get an error, and the error is totally nullified. Sir, what are the mobile phases used to herbal syrups? Herbal syrups are challenging, and in this what you wish to see is very important. If they are sugary based syrups, you are advised to use solvents like ethyl acetate, which will help you to selectively remove the organic component without the inclusion of the sugars, and that can be tried. And mobile phases, again, as I told you, it depends upon what herbal combinations are you checking in that. It is a multiple mixture. You will have to have a mobile phase, maybe a single mobile phase or plant-based mobile phase for screening of different plant compounds into that herbal product. So that is how you select the mobile phase. Anything which gives you good separation quality, toluene, chloroform, methanol, that's a very good combination and gives you a lot of bands in separation, and you can select mobile phase that is like that. How to select the slit width, we have already discussed. I will not repeat the same. If we can scan the sample spot in tungsten lamp at 520, then which lamp we can use to get the spectra? See if the molecule is visible after derivatization. Normally people ask that after derivatization I want to do a spectra. The spectra does not apply after derivatization because the molecule is already visible. It is green color, it is blue color, so the lambda max is as per the color which you see, and that is also a derivatized product, so it is converted to some other format by which you are able to visualize it. So that is why the spectrum will be not applicable over there. If it is an undervatized molecule, say any color of dye, in that case you can definitely use the spectra function where you can cover the UV and visible range with by selecting deuterium and tungsten together, and the range becomes 190 to 900 nanometers. You get a spectra of that band and you can find out what is the spectra pattern of that molecule. But derivatized molecules, we highly restrict them because they are already visible, and that is why and they are not pure compounds, they are basically converted compounds. So generally we don't recommend for going of spectras of derivatized compounds. For 6 mm bandwidth, 6 mm, which dimension should be selected? It should be around 4 into the macro and micro slit size which is there. You should select a micro. So 4 into 0.3 or 0.2 is a good slit, or even 5 would work. 5 into 0.3 micro slit is good. Then you have a question, ellagic acid and curcuminoids. I would like to point out to all my colleagues that we are pertaining this session to pharma, and that is why if your query is related to herbal, I would request you to send an email to us and we will reply, because we are right now focusing on the pharma aspect of HPTLC. So all those questions pertaining to herbal will be now answered through email. Ellagic and curcumine will reply you in an next question. Available method determination of morphine. Yeah, morphine is we have a very good method of morphine and we would like to share with you. So please, we must have already received your email ID. We'll reply to that. So with this, we end the questions which are there in the Q&A box. Few questions we'll take from the chat box and then we'll close the session. So the first question, how can we give the pre-wash? We has been explained. Gradient in TLC. TLC gradient works with development distances. So if you have a single mobile phase, you develop it with say a distance of 3 centimeter from the bottom of the plate, then you change the mobile phase ratio or some other totally different composition, and you develop it at a distance of 5 centimeter now. So from previous 3 centimeter, now you develop it to 5 centimeter. The difference which is there of 2 centimeter, you will start getting some molecules which are selective to that mobile phase over there, and that is how we establish a gradient type of development. We can go from polar to non-polar. That is a generally process. I'm sorry, from polar to non-polar to polar we go, and we can screen multiple compounds depending upon the sample. So that is how you go in gradient type of application when you are focusing on HPTLC, TLC and HPTLC. Can we replace Linomat 5 applicator with robotic applicator with the existing software? Sure, but there is a firmware requirement which is there. If you have the latest VisionCAD software, it will support, but the older softwares have a different firmware because those were designed on a different platform of Windows XP and things like that. So there might be firmware issues. So we request you to send a mail to our service department, and they will contact you and provide you an exact answer to that, whether your software will be compatible with the instrument. Pro, definitely you require the VisionCAD software. It does not work with the older softwares which are there. How to calculate theoretical plates on HPTLC? It is the number of, you have a mathematical formula in that which where you see the peak shape, and from the peak shape you calculate the theoretical plates. So you can use the same over here. Once you get the scanner, with the scanner you get the chromatograms, and from that you get the peak. From the peak you can calculate the theoretical plates. And is polysaccharide possible? Absolutely. On HPTLC, polysaccharide is possible. There are many methods, and we would be happy to share with you, and we'll share with you to your email address. And how to select a slit width, we have covered this. Establish morphine, curcumin, ellagic acid, natural dyes, spectra of visible compounds. So more or less we have covered with all the questions. Mostly they were of herbal nature, but any would like to ask any question pertaining to pharma, we'll be happy to answer them. We have two hands which are being raised. We'll just see who they are. Can we unmute them and ask them to ask the question? Yeah, Pushpalata ma'am, you have been given a chance to speak, so just unmute your mic and you can ask the question. Or Ajay sir, Ajay Chauhan sir, if you are listening to this session, please unmute yourself and we can give you the answer to your query. So you must have seen that HPTLC is being used many places as a complementary technique. Only with problematic batches, you can just go for referral checking of other using other chromatographic techniques. But for preliminary screening, preliminary checking of batches, this can be a very quick and feasible analytical tool. You have seen there are multiple dimensions in which this instrumentation is being used. So a single equipment can be used for multiple steps. You don't require any cleaning or anything of the equipment, and it can serve for the purpose of impurity profiling, cleaning batch validation, linearity, and so many things which are required in the pharma industry. It's advisable to use glass plates which give very good results for especially for the pharma concern. So yeah, Anuja ma'am.
Good morning, sir.
Good morning.
Sir, I have one question. That is, what is the difference between that Vision Cat and WinCat software? Of course, in WinCat we got the digit area, and in Vision Cat we got the area. I think that is in
Decimal
what is
points.
calculated there.
So previously when yeah, your voice is cracking, but what the question what I could make out is that the difference between the areas which you received using Vision Cats and WinCats is totally different. In WinCats, you used to get the integer in positive value, whereas in vision cats you are getting in decimal value. That is precisely because when the revision had taken of HPTLC in the vision cats, it was observed that the phenomenon is reflectance. And if you see any other spectrophotometer technique or anything, you see that it is based on transmittance. When 100% transmittance is there, you have the reading as 1, and if it is getting absorbed, you will get a reading in decimal points. Same thing applies over here, but here instead of transmittance, you have reflectance. So when you are having 100% of reflectance, that is the mirror, it will give you the signal as 1. When there is a absorbance which is taking place and the signal is not 100% which is reaching, at that time it becomes in decimal point. So this readings were then revised in, and also the 21 CFR Part 11 requirement had changed. So based on that, the newer stricter guidelines which was there, according to that, this software was made. Also, the data was made much more user friendly. You can analyze it in multiple dimensions. The quality of image, the quality of data which was being generated by the software was enhanced. You get multiple data points for analytical evaluation. That is, I can explain you, if you have a portion of this much and you want to take a reading, if you take 3 readings, you will have 3 points during this plotting of my graph between these 2 points. But if I take multiple number of readings during this graph plotting, I will get a much more accurate assumption. So these data point collection was improved in vision cats. The method library got included. You had compatibility with the Pro, which is a next generated automated system. So all these things led to the new software which was designed in with this feature, and that is why it is and much more advanced and better software. It has got all the calculations done automatically, like functions like calculations of linearity assay. You have functions of limit test, you have functions of related substances, internal standards when you are correlating it, and that happens through the software. You do not have to export the data, go through the Excel sheet and again process the data. It directly happens through the software, and one click report is generated which has directly the provision of the signature, and then it can be submitted to regulatory authorities. So all those things have been designed, and that is why it is a total different software as compared to WinCats. Hope your question is answered over here.
Thank you, sir. Thank you.
Thank you. Any more questions? Yeah, Pushplata ma'am, if you would like to ask the question. Ajay sir. Okay. Anyways, so don't worry. You can definitely write to us at lab@anchrom.in and get your queries solved. Thank you so much for being with us. Hope this session was very fruitful. And these lecture series, we have been continuously doing. I hope they are benefiting you. And if you have any kind of analytical problem or stuck up with your work, just feel free to ask us. We'll definitely provide you the guidance with that pertaining to HPTLC. With this note, we would like to end this session. May I request the marketing team to now end the session. Thank you, Sneha, so much for sharing the information to all the participants. And thank you so much, all the participants, for spending your valuable time to this session.